Here we go. So my name is Dmitry Grapov, and I'm the principal statistician at the NIH West Coast Metabolomic Center. So we specialize in both in research and fee-for-service, and so we're doing a lot of metabolomics, both targeted and untargeted, lipids, primary metabolites, glycans, and so more recently we're getting interested into incorporating proteomic and genomic measurements with our metabolomics. So as you can imagine, this leaves me a lot of time thinking about data. And so your data is not some sterile object in Excel, a block of numbers, but it's unique. It's unique to you based on your experimental design, based on the things you measured, and based on your mass spectra. So for the following presentation, I'm going to talk about some things you might consider in your data analysis workflow. And I will end with some unknown ID identification tools. So one thing you might be interested in is quality assessment. And so you might be doing this by looking at your replicated measurements and/or internal standards to estimate the analytical variance. So once you've done that and determined that your data is worth analyzing, next you're going to use your experimental design to pick up rows out of your data and test some hypothesis. Or maybe you don't know what you're looking for, and you're going to use non-supervised techniques to just identify trends in your analytes, what you've measured. Once you have identified interesting things, now you might ask the question, are these interesting things enriched within some biochemical domain? So this is a functional analysis. And finally, you can integrate all of this information, statistical, multivariate, functional, you name it, in the form of a network. And you might be doing this using a technique called network mapping, or you might know it by a different name. So let's start with data quality assessment. So here's a measurement for a single analyte. So these are replicated measurements acquired, so about 400 replicated measurements across 100 analytical batches. So these are quality control samples embedded with a far larger data set, about 5,500 samples acquired over 1.5 years. And so as you can see that we have quite a drift in the abundance of this measurement, even though it should be the same. So now that was a single analyte. Now if you want to look at this analytical variance over all analytes, you might do something like principal component analysis. And one thing you might want to do is you might want to annotate the scores of your quality controls with metadata, things you know about the experiment, like acquisition batch or time. And so you might see that, well, that actually very nicely explains the variance in your data. Now that's a big problem, because if your analytical variance is larger than your biological variance, you're going to incorrectly conclude that there are no significant differences when there really are. So that's a false negative. So that's low power. So you want to increase the power of your test. So one way you might do this is trying different data pretreatment and normalization techniques. And so one way you might be evaluating it is looking at the relative standard deviation versus the magnitude of the measurement. So in this plot, every single point is an analyte, and the different colors represent different normalization techniques. And so what you want is you want low relative standard deviation. And so you might look for techniques to decrease your analytical variance. And so I think many of you are familiar with the use of analytical standards to reduce your analytical variance. But in some cases, you cannot get standards. Maybe you're looking at such a broad data set that it's just impossible to get a good standard for everything. So in this case, you can use it using your quality control samples. So what you can do is, looking at your quality controls, which are embedded in your study, in this case 1 to 10, so every 10 samples we run a quality control, we can estimate the analytical variance. So here it is. Here's the general trend. Now that we have that, we can generate a local nonlinear model using a lowest model, and then we can model out this variance from our samples. So in black are our QCs, in red are our samples. And so we're generating a model. We're not using any knowledge about the samples. So this is purely our QCs. And so we're doing this with a little bit of machine learning, but it's relatively easy. We're really just tuning this lowest model. And so it's very important that anything you do, any normalizations you do, you should not be using any sample knowledge. That's a great way to overfit. And so you should numerically and visually validate what you've done. OK, so now that our data is solid, we're ready to do some analysis. So let's take this very simple example where we're interested in the differences between two groups. So one thing you might do is some kind of statistical test. This is great. So now you get p-values. So you know what is and what is not significant. But you're lacking rank. So something either is or is not significant. Something is not more significant than something else. And you also don't have a context. You don't know how these things are related. So you might turn to a multivariate technique, like orthogonal partial least squares discriminant analysis, OP-LSTA. Now this is a great technique for ranking all of your measurements with respect to their ability to predict differences between groups. But here again, you're lacking any significance. And again, you don't have any context. So this is where network mapping comes in. So this is a technique to combine your statistical analysis, your multivariate analysis, your functional with a context. So the network is going to be your context. And so this is very important. It's very important to look at your data from many different angles and organize it to see the big picture. And so in our group, we've been developing some tools to help you do these various analyses. So one is DVM Web. And so this is a R-based GUI. It's both web-based and local, so where you can come in, do some statistical analysis, do some hierarchical, some multivariate analysis like clustering. So it's highly interactive. And so the idea is that you can map information rapidly from what you know about your samples to these various analyses to try to learn from these analyses. And so we also have OPLS in here with appropriate model validation, and feature selection tools. And meanwhile, all of these things you can send to plotting, to our plotting toolbox, where you can visualize anything you want and map things to many various properties. Okay, so now that you've done that, you've identified what's interesting, the next question is, well, are any of these changes enriched within some biochemical domain? So this is a picture of the KEGG atlas, and so the different colors represent different pathways or domains. And so with your data, this analysis might look something like this. So essentially, the different colors just represent significantly increased or decreased metabolites. And so you ask the question, are they enriched with some domain? Now, doing this type of functional analysis is really our first opportunity for omic integration, because we can actually ask this question both with genes, proteins, and metabolites simultaneously. We asked, are the differentially expressed genes, proteins, and metabolites enriched for some pathway? And so that's actually given us more power to spot something. And so in these visualizations, we can also map genes and proteins and metabolites together. But one limitation of this approach is that we're still stuck in the pathway. And this pathway is human curated, and if there's something new going on, well, you're not going to see that. So maybe one way to get beyond this pathway is to generate networks, your own networks. And so I'm going to spend the rest of the talk talking about networks. And so a network is simply an organization of information. So the nodes are your metabolites. The edges are the connections. And so you can have different connections. You can have many connections. I'm just going to show a few. So one connection could be biochemical. Maybe this is a precursor-product relationship in some database. Another connection could be molecular fingerprint. So as the first speaker, Kai, mentioned, this is just the functional groups present in your molecule. Another great connection could be your mass spectra. And the final connection could be the connection that is dependent on your experimental design, the correlation or the partial correlation in your data. And so you're going to use this to organize your information. So here is an example of a mapped network. So this network displays the metabolic differences between control and malignant lung tissue. And so nodes, metabolites, which are red, are increased in cancer. The ones that are green are decreased in cancer. If they're large, they're more important, so they have greater magnitude. If they're outlined with a border, well, these are biomarkers in a multivariate predictive model. Their shape tells us what biochemical domain they're coming from. If there is no color, well, these weren't statistically significant, and so they're kind of out of your way. So we're using both biochemical knowledge to organize this network. Like, for example, we can go to KEGG in this case and look up that uracil is connected to 5,6-dihydrouracil. But we're also using structural similarity information. Now, this is very important for species which there is ambiguous biochemical relationships or they don't exist in any database. Like, for example, lipids. You see, there is really little biochemistry known. And so for this, we're using PubChem to essentially generate the molecular fingerprints, and then we can get a Tanimoto score for similarity. And so this score, you can actually control the threshold of connectivity. And so you can make your network sparser or tighter. And that might be something you could tune as well. So previously I showed that we had different changes occurring, and so some of them can be explained because they're occurring within the same biochemical domain or among structurally similar molecules. So for example, if we look over here, we see a lot of nucleotides are going up together, so that might be explained. We see a lot of our lipids are actually going down. But the next question could be, well, are the changes in lipids and nucleotides related at all? Or maybe are the changes in our nucleotides and amino acids, are they related? How are they related? So how do we explain that? So to do that, we turn to our empirical network. This is our empirical edge. So what you're looking at now, this is a partial correlation network where if something is connected with an orange edge, it means it's a positive linear correlation. If it's a negative edge, that means it's an inverse correlation, as one thing went up and the other one went down. And so again, we're using this idea of network mapping to visually guide you through this network. So our most important molecule is, well, in the center. And this wasn't organized by me. This is just data-driven. So it's big. It was a big change. It was important on a multivariate level. And it also has the most connections. So it's kind of drawing our eyes there. And so we can look at it, and well, we see that it's actually reporting on nucleotide synthesis. So we actually have 3 different markers of different nucleotides going up. So in cancer, DNA synthesis is up, nucleotide synthesis is up. So next, we can look at some of our amino acids. Look at this cluster, they all have the same shape. And so we see that the increase in nucleotides is negatively correlated to a decrease in amino acids, and specifically 2 urea cycle species. And it's converted through glucose, whatever that might mean. And then we can continue this, and now look at this module of changes over here. And well, looking at these, a domain expert can tell, well, this is an increase in protein glycosylation. Nucleotides went up, urea cycle species went down, protein glycosylation went up. So that's interesting. But oftentimes, what we see is that it is the unknowns that have the most interesting changes. So we do large experiments, and then our unknowns are the best biomarkers. And so the first 2 speakers spoke about the difficulties of unknowns, and so I'd just like to take the next couple of slides to think about some maybe alternatives, some quicker, maybe easier techniques. So Peter Dorstein's group very eloquently showed that you can take a bunch of MSMS spectra and organize all of this spectra in the form of a network, a mass spectral similarity network. So I saw this last time at ASMS, and I loved it, and I immediately incorporated it into my own routines. And so here what I've done is I've generated a structural similarity network of all my known compounds. And then I used the mass spectral similarity of my unknowns to connect them to my knowns. So this is electron ionization spectra here. And so now I can mine this and try to get a better idea of maybe what biochemical domain these species are coming from. And so for example, these 2, I see they're kind of spanning the range between sterols and fatty acids. Well, and they have the same color and shape as actually 3 phytosterols. So in my system, they're changing in the same way. And so I might conclude that these could likely be phytosterol esters or sterols hooked to a fatty acid. And so that could help me constrain my database search. Okay, so we can do that with structural similarity and mass spectra, but we can also do that with empirical correlations. So in our group, we're very interested in diabetes, both type 1 and type 2. And so over the course of many studies, we keep seeing certain unknowns, which are very interesting. They're very good biomarkers for type 2 diabetes, and we would love to know what they are. And so when I model this, we can look at the correlations, and so I often see that these species are highly correlated to glucose. And not only that, they also show very similar patterns, even for 2 different groups for glucose. This is a time course. And these species are also correlated to each other, and their mass spectra match glucose very well. So I would conclude, well, this is likely a carbohydrate, something very similar to glucose, and it's really telling me the same signal as glucose. So as a statistician, I wouldn't really think of this as very interesting. But later, we went on through many rigorous chemical experiments to identify these unknowns, and lo and behold, this is glucose. So we're doing GC-TOF here. We were doing a derivatization with TMS, and so this is just partial derivatization products of glucose. So kind of a problem solved here. So in our group, we're also developing a tool to help you generate these types of networks, so MetaMapper. And so it's also integrated with our chemical translation system, CTS. So you can go from name to various database identifiers, which are then going to help you search the various databases. So over 200 of them, so you might then go to KEGG and calculate your biochemical relationships, maybe generate your network. Here's one for the TCA cycle. Then you can quickly add your structural similarity information to this, just using the identifiers, PubChem lookup. Now you can upload a real data set with mass spectra and data, create a spectral similarity network, rapidly add biochemical and structural similarity information to this network, and then add partial correlation and correlation information to all of this. And so you're kind of in control. So the possibilities are kind of really endless. And so one thing that showed is that as you're adding all of these different ways of connecting your information, you're kind of giving a context to everything. And so the various relationship, structural similarity, mass spectral, they all have weights. And so you would go on to build a network, and in this network, you could map the strength of the relationship, how much you believe it, maybe by the thickness of the line. So you would orient yourself among the network to things which you believe less or more, given the data. So what I've shown you is kind of some of the things you might be thinking about as you're doing your data analysis, your quality assessment, your statistical, functional analyses, and finally ending at network mapping. And so this is a powerful technique because it's really independent of pathways. All you need is a database, and really, you can combine pretty much anything. So here, this is a combination of over 1,200 different metabolites. This is some of the many known species we measure And so one thing you see is, well, we know where lipids are in the context of many other things like organic acids, amino acids. And so you don't often see this. Lipids are actually, it's actually very difficult to combine lipid information with primary metabolite information. This is because there is very little database knowledge about their connections. And so here we're using structural similarity. We could add spectral similarity, and we can go on and see how these species are linked. And so, as I mentioned when I started, one of our interests is to go on and integrate both proteomic and genomic information into these networks. And so there are a variety of tools out there to do this. Really, all it takes is a database. Because where before we had a straight connection between two metabolites, now we just insert an enzyme. Now, the metabolites are transformed through an enzyme, and then we add the mapping of what genes encode for this enzyme. And so now we've incorporated our proteomics and genomics information into this. So everything I've talked to you about and more is completely freely available. It's all written in R, so many of it has GUIs. And so I'd just like to thank all the American taxpayers for funding this, so it's available to everybody. And so if you have any questions, feel free to email me, or if you're interested about the NIH West Coast Metabolomic Center, we do offer fee-for-service data analysis, if that's something you're interested in. So you can contact us at metabolomics@ucdavis.edu. And I'd like to acknowledge my funding, NIH, and thank you for your attention.